Hi, everyone. Thanks for attending today and joining us. My name is Caroline Storr. I'm a senior scientist at PacBio, now on the customer experience team for software and informatics. And today, my presentation is going to be brief. I'm just providing an introduction to some of the PacBio software tools that you're going to be learning about today from Nancy, and what they can do beyond QC. But first, I sort of want to bring up why talk about QC in a bioinformatics webinar at all. And that's because all bioinformatics begins with undoing your sample from the input tissue and how you've collected it to sequencing and the run, which is what we're going to be doing a deep dive in today. So just keeping that in mind when going into data analysis. Are you working with cells, whole blood, bacteria, tissue, what kind of tissue, plants, and what other microorganisms might be coming along for the ride. The software that we're going to be talking today is called SMRT Link, specifically version 12.0. And this is PacBio's software that's used by primarily instrument owners for setting up samples to prepare them for sequencing, run QC, which is what we're going to be talking about today a lot, data management, and also optionally some secondary analysis. And so the software what you see here is an example of the graphical user interface, but there's also command line tools that come with the software itself. And there's APIs for integration into, for example, your LIMS systems. So where does this software exist within the whole PacBio sequencing systems, right? So here's your instruments that you might provide or the customer might have. And then this instrument communicates with your SMRT Link server here. And you access this server through your browser computer, in this case, a Chrome browser, and which enables you to see your sequence data, which is stored on the storage server here. The SMRT Link software has many modules for managing both your instruments, again, like I mentioned, sample setup, setting up runs, managing data, and also secondary analysis. I'm not going to go into depth too much in each of these today, but I just, so you're familiar with your options, wanted to mention what's in SMRT Analysis. There are two types of analyses you can perform. One is analysis jobs that are designed for biologically meaningful, to get biologically meaningful results. And then this includes things like mapping to your reference for, say, variant calling, analyzing transcriptome data using PacBio full-length transcript isoseq analysis, microbial genome analysis, which includes genome assembly, as well as 6-methyl-A and 4-MC detection. Our newer workflow for read segmentation and single-cell transcript analysis called isoseq analysis, as well as a new workflow for variant calling based on deep variant for small variants and PBSV for structural variants. There's also some data utilities included in this analysis, including 5MC calling, if you haven't already done that on instruments, which is the most common place 5MC callings happen, demultiplexing using our tool Lima, as well as some miscellaneous utilities like generating FASTA and FASTQ files, using export reads, marking PCR duplicates depending on the type of sample and data you have, undoing demultiplexing, turning a MOS array sequence into the S reads that it's comprised of, and trimming ultra-low adapters, which is part of our ultra-low prep. So those are some of the options you have available to SMRT Analysis. And so where can you find SMRT Link or SMRT Analysis or alternatives to this if you just want to use the command line and don't want a full weight tool for yourself? So you can find information on the SMRT Link software if you go to PacBio downloads and installation, as well as the compute recs requirements for it. If you want the same tools available to you as a package, but only in the command line, there's also a SMRT Tools installation, which is the same tools from SMRT Link, but only the command line version of them. And then if you want to just pick and choose tools as you need them, a lot of those, especially analysis tools, but also data utilities, are available on Bioconda as well. Today we're doing a deep dive into QC in particular, so I just wanted to spend a minute on how you actually get your QC data from SMRT Link or from your service provider if you're sending your samples out for sequencing. And so in the data management module, you go to you look at all your data sets, you click on a hyperlink to a data set, You'll see a bunch of options, including the path to your data, some thumbnails for different graphics, as well as metrics. And so this is called your data set report. And so this is all, you'll see these figures again, as Nancy will bring them up. And you can actually export this as a PDF. And so here's an example of how you do. You export, check the box for export PDF reports. You don't need to include the data set, and then you're going to get this downloadable PDF. So you want to make sure your pop-up blockers aren't preventing that. And so, you know, as you think about what Nancy shares with you today and about your next sequencing run or past sequencing runs, how you get access to the same data, well, especially if you don't own an instrument yourself and don't already have SMRT Link installed, well, you could request this PDF data set report with your sequence data from your service provider. So that's no additional software required. Probably the lighter weight option, if you do want your own tools, is to go with the command line only option of Smart Tools. However, you are going to require most instrument output files from that. And so you'd install Smart Tools, and then the tool you want is Run QC Reports, the command line tool. And then the third option, it requires, again, most of the instrument output files, and that is installing SMRT Link on a Linux server with the required compute. And then you can just import your data via your run data into data management using the graphical user interface. So these are sort of your options from most lightweight in terms of what you have to install to, to largest. So before we begin, just some quick take-home messages. Just remember, bioinformatics begins with QC, which is why we're doing a deep dive into QC today, that you can get these QC reports through the SMRT Link software, either by installing it yourself or by requesting the PDF for their service provider. So without further ado, I'll hand it off to my colleague, Nancy.
Okay, so for about the next 30 minutes or so, I'm going to talk to you. So after we look at the output files and directory structure, then we'll take some time going over our smart sequencing metrics terminology so we get familiar with the terms that we'll be using to determine how well your sequencing run went. Then we'll go over the SMRT Link Run QC module. And then we'll take a deeper dive into the run details report metrics summary table. We'll take a look at some of the visualization plots. And then finally, I'll leave you with some 3 questions you can ask yourself to help interpret these run QC results to assess the quality of your run. Okay, so let's talk about the on-instrument analysis workflow pipeline. So at the top of the screen, you see a pink chevron that describes the entire workflow, starting from your sample prep through library preparation and sequencing and so on. So we're going to start over here on the right on the on-instrument data analysis. So there are 3 different computers that are actually on the Revio system. One is called the real-time or RT computer. And this is where sequencing data collection and primary analysis data processing is occurring. So that's actual base calling that's happening during the sequencing in real time. Then there are 2 other computers called NRTA and NRTB. These are non-real-time computers. And they're responsible for the post-primary analysis processes and the on-instrument secondary analysis, which includes circular consensus sequencing, or CCS, which is used to generate your HiFi data, 5MC calling, and demultiplexing by barcode. And so once these processes are complete, the data is then transferred off of the Revio and onto your storage and SMRT Link server. All right. So during primary analysis, a real-time base calling pipeline is run actually during data acquisition. So while the sample is sequencing inside of a SMRT Cells EMW. And during primary analysis, a single processing algorithms are actually converting those raw photon counts into base calls. And then following that, post-primary analysis, which is occurring on those NRTA and NRTB computers, this is happening after data acquisition, so after a 24-hour movie completes. And it basically consists of 3 different components. One is a high-quality region finder algorithm. We'll actually go through and annotate high-quality regions of a long polymerase read or a raw sequencing read. And it will trim out those low-quality regions that happen at the beginning or early sequences of a polymerase read. The second component is there's an algorithm that will go in and find the adapter sequences on each of the SMRT templates, annotate them, and filter them out of your data set. And lastly, the spike in DNA internal control is identified, annotated, and then also filtered out of your data before delivery. So after the post-primary analysis completes, there's a series of on-instrument secondary analysis that is also happening. So the first thing is something called circular consensus sequencing, or CCS, which is really a process of taking the multiple polymerase passes on a single molecule and performing this intramolecular error correction to generate one circular consensus read per molecule. And then those CCS reads get a predicted read quality score, and if that quality score is at least QV20 or 99% accurate or higher, it's called a HiFi read. And any CCS reads that fall below that metric gets binned into a category called fail reads. So on both bins, a tool called Jasmine is then called to do your methylation calling at CPG sites for 5MC. And using the kinetics of the polymerase as base rate incorporation, you're able to find the F5MCs at those CPG sites, and then those that information is stored as BAM tags in the output files. And then secondly, a tool called Lima is then called to demultiplex your samples according to barcodes. And so for each HiFi read or sample, you'll have a separate HiFi read BAM file. So once those files are generated, it is then removed off of the Revio instrument and transferred to your storage and SMRT Link server. Okay, so let's take a look at the output files and directory structure you could expect off the Revio. So like I mentioned, the Revio system will automatically generate two types of data output files. You'll have your HiFi reads and your fail reads. And for each barcode after demultiplexing, you'll get a separate HiFi read BAM file and a separate fail reads BAM file as well. And so all reads from both bins will have annotated 5MC methylation calls at those CPG sites, and those will be saved as MM and ML tags. And additionally, if you had selected during run design to keep your summary metrics, you'll also have a series of tags listed here that will be included in each read's output BAM files. Okay, and so if you were to look at an output file directory structure, then the name of the directory is going to start with an R, meaning run, followed by a couple digits, which is usually a serial number of the Revio instrument the run was sequenced on, followed by the date and time. And in that directory, there are a series of subdirectories that are representing the individual SMRT cells that were part of that run. And so the annotations A01, B01, C01, and D01 actually represents the well in a 96 well plate that your sample was actually put onto the instrument. So if you were to click on one of the sample subdirectories, let's take A01 for example, there's then another series of subdirectories where you can find your fail reads, HiFi reads, metadata files, PB format files, and other statistics. And so you could essentially have a service provider can just make a giant tarball of this run directory and send that out as delivery to your customer. And then you could just direct your end user to the HiFi read subdirectory for downstream data analysis. Okay, so now let's take some time to go over smart sequencing metrics terminology and get better acquainted with this dictionary. Okay, so let's consider a SMRT Bell template, which is really a linear double-stranded DNA template that is bookended on either end by our hairpin adapters. And it's on these hairpin adapters that a sequencing primer is annealed to so that a polymerase, sequencing polymerase can then be bound to the complex. And during the course of a sequencing run, the polymerase will open up the template, which will become topologically circular from the perspective of the polymerase. And the polymerase will read the adapter sequence followed by, let's say, the reverse strand here in purple of the DNA template, followed again by the adapter sequence in green, and then the forward strand here in yellow, and then again the adapter sequence, and it will repeat this over and over again throughout the 24 hours of a movie. So the result of that sequencing reaction is a very long, raw, unfiltered read. And actually the early sequences at the start of a movie collection contain low quality base calls. And so one of the first processes of the post-primary analysis is to find this low quality region and trim it out. And once that low quality region is trimmed out, you're then left with what's called a polymerase read. The polymerase read will still have your adapter sequences in it, sandwiched between the single passes of the forward strand followed by the reverse strand, adapter, forward, reverse, and so on. And so the polymerase read actually represents the total number of bases produced from a ZMW after trimming out that low quality region. So it is going to be reported in your run report metrics, and it is a QC measurement that we'll discuss further later in this talk. Okay, so now you have a polymerase read, it still has adapter sequences in it. So the next step during post-primary analysis, actually it's a secondary analysis, you will then trim out the adapter sequences, and then when CCS is called, your circular consensus sequencing algorithm, it will stack all of these subreads up and then do intramolecular error correction on those subreads to come up with a circular consensus sequence or CCS read per molecule. So as you can see here, subreads, there are going to be multiple subreads per single molecule, which represents individual passes of the polymerase along the template. So any CCS read that has a predicted quality score of QV20 or higher is going to be called a HiFi read, and anything falling below that threshold is just a fail read or other CCS read. Okay, so I like this summary table to keep all of our definitions straight. So on the left here, a polymerase read, remember, has all of your subreads plus your adapter sequences. And the high quality region is the only part that you'll see in a polymerase read. The low quality region has been trimmed out. And so for every single molecule that's sequenced, you'll have one polymerase read. And like I mentioned before, this is also used as a sequencing run performance QC that we'll talk about in a little bit. And as a reminder, subreads represent individual passes of both the forward and reverse or positive negative strand of your DNA template. Some of them can be truncated because it represents a part in the template that the polymerase stopped sequencing. So it gives you a somewhat of a sense of how big your library insert is, but it will be off by a little bit. And so remember, each single molecule will have multiple subreads. And then finally, CCS reads will represent your intramolecular consensus sequence for an insert. And here again, one single molecule will have only one CCS read. And CCS reads can be used as a sort of sanity check to make sure that the CCS read length distribution closely matches your library's insert size QC. And then finally, your HiFi reads are just CCS reads with a QV20 or higher, or 99% accuracy or higher. And so one molecule will only have one HiFi read. And so you'll want to use your HiFi read length distribution for your downstream data analysis applications, or not just the distribution, sorry. You'll want to use the HiFi reads for analysis, but you want to look at the HiFi read length distribution as a way of checking or confirming your library insert size QC. Okay. Another set of very important smart sequencing metrics definitions concerns productivity. So productivity is a measure of whether or not a SMRT cell ZMW actually produced useful sequence read data. And so ZMWs sort of fit among three productivity bins. So your P0 or empty ZMWs represents ZMWs where no read data was produced. So essentially no signal was detected. A P1 or a ZMW that's productive is a ZMW that's produced useful sequence data. You were able to get polymerase reads out of it. And typically this means there's one actively sequencing template polymerase complex bound to the bottom of the ZMW. Then the last bin is called P2 or other ZMW, and this is a ZMW where no useful sequence data was generated. Typically, this is due to multiple template polymerase complexes being bound to the bottom of the ZMW, and this creates a lot of background noise, and base calling is not being able to be done appropriately. And so this brings us to a really important concept of optimizing sample loading onto the SMRT cells in order to maximize the amount of smart sequencing or HiFi data you're able to get. So when a sample is overloaded on a SMRT cell, this will increase the chances of a multiply loaded ZMW. And in that scenario, you get a very high background noise and overall lower data quality and shorter polymerase read lengths. And so some definitions of P0, P1, and P2 that will help you determine whether your run was underloaded or overloaded are as follows. So an underloaded SMRT cell is actually defined as having a P0 of much higher than 40%. And in this scenario, most ZMWs are not producing any read data at all. An optimally loaded SMRT cell will have both a P0 of at least 20% or more and a P1 or productive ZMW percentage of about 50 to 70. It really depends on insert size. And in this scenario, most ZMWs will produce useful sequence data. In the scenario of an overloaded SMRT cell, the definition here is actually a P0 that's much less than 20%. And in this scenario, most ZMWs are not going to be able to produce useful sequence data due to this overloading and crowding of the ZMW issue. All right, so now let's take a look at the SMRT Link Run QC module. So when you go into your SMRT Link, you'll want to navigate to the Runs module, and here you'll be able to look at your run metadata overview and a summary of the consumables used for your particular sequencing run. You'll also be able to look at the smart sequencing metrics table for both your sample and the internal spike in control metrics. So things like productivity, like we just talked about, and your HiFi data yield, and so on. And then also you'll be able to look at your run detail report visualization plots. And this will give you a quick way to assess the quality of your library and did it match your expectations. Okay. So once you click on the runs homepage, you'll see a sort of Excel-like table where on the left-hand side under run name, you'll see your sequencing run that you could click on and if you see in the run status column that run was complete, you'll be able to click on it and assess the quality of your run. So when you open that view and when you click on a run, at the very top, you'll actually have 4 different sections that will be displayed. At the very top, you'll have this run metadata summary table. And here you'll see things like when the run was started, how many SMRT cells were part of that run. You'll see the instrument serial number, the instrument control software versions, and so on. So this is all very useful information. If you ever do have a problem and need to reach out to tech support, they'll want to see this information. In section 2, that's actually not expanded in this view, is a table of your consumables that were used during sequencing. So this includes the sequencing reagent, lot numbers, expiration dates, as well as the SMRT cell lot numbers and expiration dates. So in the middle here, you'll have a section for your primary analysis metrics summary table that we'll take a deep dive into momentarily. And underneath that table, you'll actually see a 4th section of a bunch of thumbnail views of some visualization plots that we'll get to later as well. Okay, so let's take a deep dive into the summary table. So of course, the first thing that you're going to want to look for and draw your eyes towards is your data yield, right? So in blue highlighted here, one really important column you'll look at is called total bases in GB, and this is actually your P1 ZMWs times the polymerase read length, and that represents the total amount of data you're getting off of your run. The second most the quickly viewed column is your HiFi base yield in GB. And so this is the total base yield of CCS reads with a QB score of 20 or higher. And the HiFi data yield really depends on DNA sample quality, insert size, and loading concentration. Okay, so then the next table, or the next section of the read metrics table, I should say, that you'll want to look at is your productivity that we just discussed. So it'll be listed as percentages of ZMWs that are P0, P1, or P2. And so P0, as you remember, is an empty ZMW where no signal was detected. And the percent P1 is your productive ZMWs with a high quality, where a high quality polymerase read was generated. And then the P2 is other ZMW where a signal was detected, but no high quality polymerase read was able to be generated. And so looking at this table, you'll want to assess, was P0 values, less than or greater than 20%? Was P1 within 50 to 70%? And this will give you a clue into how well the SMRT cell was loaded. The next part of the table that you'll want to take a look at is you'll want to expand this section called reads. And here underneath, you'll see data points for your HiFi reads. And the first column will be the number of HiFi reads, which are again defined as having a QB score greater than 20. And so in this example, you see 6.4 million HiFi reads. And in the next column, you'll have your yield in GB. And in the third column, you'll have the mean length of your HiFi reads, and this should roughly correspond to your expected library insert size. And then lastly, we have a column of the median QB scores for the reads in that particular sample. Okay, so the next section of the summary table you want to look at are the polymerase read metrics. And so the first column will be the mean polymerase read length. And for a typical 24-hour movie on the Revio, you're expecting a polymerase read length mean of at least 60 kb or more. Any polymerase read lengths that are below 60 kb could start pointing to some potential sample issues like sample contamination or suboptimal loading, or a combination of both. So that's one metric that you for sure want to check right away. Are my polymerase read lengths at least 60 kb or more? The second column is the N50 of the polymerase read lengths, and for a whole genome library on the Revio 24-hour movie, you're hoping for an N50 of at least 120 kb. Okay, so if you take a look and expand the control section of the summary metrics table, you'll see a couple of columns that will give you metrics of the internal spike in control. And so the control that you actually spike in is this synthetic 11 kb construct that has a known sequence. And so during the post-primary analysis, it can be very easily separated from the rest of your data and analyzed and reported back here in this metric table. So similarly to the sample metrics, we're also going to be calculating the polymerase read lengths, or the mean polymerase read lengths of the control. And so for the control, you want to see at least 50 kb for a 24-hour run on the Revio. And the total number of reads, you should be expecting at least 750 control reads coming out per sample. And then the last two columns are concordance. So we're reporting the mean and mode concordance. And typically the values should be greater than or equal to 0.88. Okay, so next section of the summary table that you'll want to take a look at involves the local base rate. So the local base rate refers to the average base incorporation rate of the sequencing polymerase during sequencing. And so the Revio polymerase usually takes about 2 to 3 bases per second. And so that average is going to be reported here. And again, if the local base rate is below this expected rate or much below this expected rate, you could start troubleshooting some issues with sample quality, potential contaminants, and so on. And so under template, there are 2 more columns called adapter dimer and short insert. And these values should ideally be below 2%. Okay, so let's take a look at some of the report plots. So one of the first plots that folks take a look at are the polymerase read length plots. And so on the left here, I'm showing you a 19 kb human whole genome library on the Revio. And on the right, a representative 14 kb MAS-seq single cell library. And so the first thing you'll notice is a high peak on the left-hand side. This represents polymerases that terminated very early on during the 24-hour movie. This large peak is expected. It's not a cause for concern. It's a known phenomenon. And then you'll see the read lengths going up to 250 kb. And so taking the mean of both the entire output, typically you'll have a polymerase read length of at least 60 kb or higher. And you'll see that you'll never see too many reads beyond 250 kb because remember, you're limited to a 24-hour movie, and the polymerase has a known base incorporation rate of 2 to 3 bases per second. So it's very normal to have a drop off at 250 kb. And sometimes you'll even see a pileup of reads at 250 kb, and this represents polymerases that only stopped sequencing because the movie stopped at 24 hours. Okay, so a next plot that you'll see is the base yield density. So again, on the left, I'm showing you a whole genome library, and on the right, a MAS-seq single cell library. And here, your quick visualization of these plots, you should look for an N50 of at least 120 kb for a 24-hour movie. Okay, so the next plot is called the read length density plot. It's essentially a heat map where on the x-axis, we're plotting the polymerase read lengths, and on the y-axis, we're plotting the process read length. So it's an approximation of what the library insert size should be. So you'll see, and in red represents the highest concentration of your reads. And for a whole genome sequencing library, most inserts tend to be between 10 and 30 kb. So this area of red should very closely match what you're expecting your library to look like. And then for a MAS-seq library where the inserts are usually a very tight distribution, typically around 14 to 16 kb, you'll see a concentration of red in that particular area. So if the density of reads is far away from the size that you expect your library to be, it could point to some sample issues. Okay. And so you also have a series of HiFi data specific plots. So on the left, you'll have a histogram, essentially the HiFi read length distribution. And the mode of this distribution should approximately correlate to the physical library insert size. In the middle here is the read quality distribution plots. Again, the histogram of the overall predicted accuracy scores of each of the reads in your sample. And you'll see a peak here of QB or Q50 or higher, and this is usually due to any residual short insert fragments that get sequenced many, many times over the course of 24-hour movies, and you get this pileup here. But typically, you'll see most reads being between Q25 and Q30 for a whole genome library. And then finally, you have another heat map visualization plot that is looking at your predicted accuracy versus your read length. And again, the red represents the highest concentration of where most of your reads are falling on that distribution. Okay, and lastly, we'll talk about the 5MC detection plots. So this example here is for a human whole genome library. And so you can think of the 5MC methylation status at a CPG site as expressed as a probability of being methylated or not based on our statistical model. So a probability of 1 will indicate that a CPG site is most likely going to be methylated, and a probability of 0 indicates that the CPG site is deemed unmethylated. And so for a human eukaryotic sample where you expect a lot of CpG methylation, you'll see a sort of rightward skew towards a probability of 1. And as a comparison, here's a plot of a non-eukaryotic sample where we don't expect there to be a lot of CpG methylation, and you could see a strong leftward skew towards 0. So that's basically how you want to quickly interpret these plots. Okay, so finally I want to end on a summary slide of your major takeaway messages from looking at the Run QC summary plot, or table rather. So one question you want to ask yourself when you evaluate your sequencing performance results is, do the primary run metrics indicate that my library was successfully sequenced, right? So the first thing you want to look at is your total bases output, your productivity percentages, do they fall within the expected range for an optimally loaded SMRT cell? And what is my mean polymerase read length? You want to make sure it's at least 60 kb or higher. If it's lower than that, it may point to some troubleshooting you need to do on the sample prep side. And then secondly, you want to take a look at your DNA internal control, and are those metrics as expected? So you want to check, do you have at least 750 control reads? Are the mean polymerase read lengths of your control reads at least 50 kb or higher? And is the concordance of those control reads at least 0.88 or higher? And so some of these metrics are going to be one of the first things your local FAS or tech support crew will look at if you reach out and asking for some guidance on your sequencing performance run. And so then finally, you want to look at your HiFi data specifically, and do the sequencing data properly reflect what your expected properties of your SMRTbell library are? So does the mean HiFi read length distribution look like your library QC? And you could check that by looking at several of these plots. And again, if the sizes don't quite match up, then you know you have some troubleshooting to do. So with that, thank you for your attention.